Hello everyone, welcome to Smita Kiran Biology classes. In this video, let us discuss the transcription unit and the concept of gene and what is gene, structural genes and all. Followed by the transcription in prokaryotes. When we see the structure of DNA, it consists of a unit that undergo transcription called transcription unit. It is characterized by the presence of promoter sequence, terminator sequence and also the structural genes. Now let us understand the difference between the genes and structural genes. Gene in chapter inheritance we have studied, it is the unit of DNA that is responsible for the inheritance. That is, the functional unit of DNA or inheritance is called the gene. In terms of DNA sequence, the sequence of DNA that produce mRNA, tRNA or rRNA is called as a gene. Now, what is meant by structural gene? We can call structural gene as a cistron. Invariably, you can use the term cistron or a structural gene. Structural gene is one that can synthesize a polypeptide chain upon the process of translation. That means from dRNA, mRNA is produced, from mRNA, protein is synthesized. Now, these structural genes or cistron that is coding for polypeptide is of two types. One is monocistronic gene that code for a single polypeptide as it is seen in case of eukaryotes. When we see the DNA sequence or the structural gene, that structural gene will code for only one polypeptide, whereas polycistronic gene code for more than one polypeptide as in case of prokaryotes. Now, in case of eukaryotes, the monocistronic genes are not continuously coding for an entire polypeptide together because they are called split genes. Because if we see here, if it is a DNA mRNA sequence which is synthesized from a template DNA strand upon transcription, it have a coding sequences and non-coding sequences. Coding sequences are called exons and non-coding sequences are called introns. Now, the exons, the coding sequences will appear in the matured or processed mRNA, whereas introns did not appear in a processed mRNA, which means what? Only exons are possible or responsible for the synthesis of polypeptide upon the process of translation, whereas introns are removed from mRNA during the process called splicing. And remember, this is only applicable to eukaryotes, whereas the genes are continuously coding for the protein in case of prokaryotes. Now, if the sequence of DNA, if we take, it have a promoter sequence and a terminator sequence, and also before and after there is presence of non-coding sequences. Now, the sequences of promoter and the terminator sequence also affect the process called inheritance, because these sequences are responsible for regulating the inheritance and they do not appear in RNA or protein. This point is very important. You should remember. Now, let us understand the process of transcription and types of mRNA in prokaryotes. In prokaryotes or eukaryotes, whatever, there are three types of mRNA produced. mRNA, which provides a template for the synthesis of amino acid sequence. tRNA, it brings amino acids and read the genetic code which is present on mRNA. And rRNA is structural and catalytic in function, and all these will work during the process called translation, that is the synthesis of protein from mRNA. So here there is a requirement of all the three types of RNA, mRNA, tRNA and rRNA. Now let us understand the process of transcription in prokaryotes in detail. In case of prokaryotes, the transcription process we have studied already. Synthesis of tRNA, mRNA or rRNA is called transcription, right? So here all three types of RNA molecules are synthesized from a single type of enzyme, that is DNA dependent RNA polymerase. and this process will happen in 3 steps: initiation, elongation and termination. If we take a sequence of DNA that has to undergo transcription, here it is having 2 terminals, that is 3' to 5' and 5' to 3'. And we know already that 5' to 3' is called a template and 5' to 3' is called a coding sequence. Now, RNA polymerase enzyme will bind to a promoter sequence. In case of prokaryotes, the promoter sequence is TAT-AAT. It is called a Pribnow box, to which a RNA polymerase enzyme will bind, and that will initiate the process called transcription. Now, it comes elongation. For elongation process, there must be a association of RNA polymerase with a initiation factor. That means this initiation factor will initiate the process called elongation of ribonucleoside triphosphate, which are acting as a substrates. Now remember, elongation is initiated by association, a transient association of sigma factor with RNA polymerase. Now this RNA polymerase will keep a loop of a double strand which is converted into single strand, so that the process of transcription will take place continuously, and also there is a association of RNA with this ribosome, and that RNA is called nascent RNA. Now when a RNA polymerase enzyme polymerizes entire structural genes and reach a terminator sequence, the RNA polymerase is get associated with a terminating factor that is called Rho factor. At this time, RNA polymerase and also nascent DNA will fall off from a transcription unit, and that will result in the process called termination. By this time, there is an end of transcription. In case of prokaryotes, the process of transcription and translation are coupled. For this, there are 2 very important reasons. The first reason is the functional mRNA doesn't require any additional processing. That is, in case of eukaryotes, there is a process called splicing, capping and tailing, and all these processes are not at all required for the maturation of mRNA. So that is the first very important reason. The second important reason is, in prokaryotes, there is no differentiation of cytosol and the nucleus, because transcription in eukaryotes takes place in nucleus, translation takes place in cytoplasm, but there is no division, that is compartmentalization is not there in prokaryotes. Hence transcription and translation takes place together. That is, much before the completion of RNA synthesis, there is a synthesis of polypeptide chain. In next video, let us discuss transcription in eukaryotes. Thank you.